As Maria said, my name is Luke Smith. I'm working in Dr. Plaxco's group under the mentorship of Dinah Simon as part of the Beckman Scholars Program. So my research focuses on the area of the development of new smart materials, which are materials that are capable of responding to changes in their environment. Now, the great thing about smart materials is that they have the potential to completely revolutionize drug delivery. Now, these materials already exist to a certain extent. For example, liquid gel capsules that are capable of responding to the low pH of your stomach to dissolve there and release their payload. But a change in pH is a pretty drastic change. And if we want to make better smart materials, we have to make smart materials that are capable of responding to more subtle cues that could arise from a range of conditions, for example, like inflammation, all the way to cancer, hopefully. And so, so if smart materials are the way forward, the question is, how can we make these materials that respond to these specific molecular cues? So the Plaxco Lab's approach to this is to use biopolymer networks to make these types of responsive materials. And the reason we're able to do this is through aptamer technology. So aptamers are essentially short, single-stranded pieces of DNA that are capable of recognizing and binding to specific molecular cues. And what we've done is we've taken the aptamer and placed them into this larger polymer that we like to fondly call the super sandwich, which is composed of alternating strands of aptamer and structural strands, as being seen in red and blue. Or with this system, there's an inherent trade-off between the stability or strength of the polymer and its responsiveness that comes about as a result of this overlapping region here. So the specific goal of my research is to optimize this trade-off between strength and responsiveness. And so in order to monitor the system and see which direction our, I'm optimizing this by altering the overlapping regions of the super sandwich polymer. And so in order to monitor whether these changes are having a good effect or a bad effect, we have to actually look at the system. The way that we do that is by labeling the aptamer strand with a fluorophore here and a quencher on opposite ends of the aptamer strand. And the fluorophore and quencher, when they're far apart, when the super sandwich is intact, this gives us a fluorescence on signal. However, when we introduce the molecular cue, it will bind the aptamer and form this stem loop, bringing the fluorophore and quencher close together and giving us a darker signal off. And so, in order to accurately determine the kinetic rates that this happens at, we need a system, a piece of equipment that is capable of taking fluorescence measurements thousands of times in almost 1/100th of a second, and that's where the stop flow fluorimeter comes in, which is a rather specialized piece of kit. And so, with any novel system like the super sandwich, the first step is characterization. So the first experiment we did was to figure out the relative concentrations of the aptamer strands and structural strands we needed to actually form the super sandwich. And so after determining the optimal ways to form the super sandwich, which we figured out based on this titration experiment, the next step was figuring out just how sensitive it is to our molecular cue, and for our model system that cue is ATP. And so in this experiment, we titrated in ATP and found that at about concentrations of around 7 millimolar ATP, we would get about a 50% signal reduction. And from that point, we wanted to see just how fast is this response actually happening. And so in order to see a kind of ceiling value for that, we decided to use 10 times the concentration for 50% signal reduction, which would be 70 millimolar ATP, and we got this nice curve which shows that the super sandwich actually responds in under 2 seconds to those concentrations of ATP, going from its polymerized form here down to its bound form. And we thought that was a pretty good result. So in summary, for the super sandwich, we're able to characterize the system, determine experimental parameters. I know it's almost lunch, guys. And overall, this kind of served as a proof of concept for the system, allowing us to just check that it actually works the way that we hoped it would. So having completed that goal, we decided to move on to a slightly more challenging aspect and essentially extrapolate the super sandwich system into a more interesting one, the hydrogel. So the hydrogel works on the same principles as the super sandwich system, except now the structural support strands are actually these red and green tips on these pieces of Y-shaped pieces of DNA, and the aptamer strand is this little black piece of DNA here. And these come together to self-assemble into a kind of honeycomb-like structure and form a hydrogel, and hopefully they will break apart in the same way as the super sandwich. And so with this system, the first step was to actually make these monomers, which is slightly more challenging than we would have thought. So the way that we do that is we take the original single-stranded pieces of DNA, put them in a PCR tube, and put them into a thermocycler here, heat them up to 95 degrees and Celsius, and then slowly cool them back down with the hopes that this will form monomers. However, the trick is the cooling rate is very specific, and there's a lot of optimization that can happen there. So we tried a bunch of different cooling rates, three of which are shown here. We have two gradient cooling rates, a 2-hour and a 4-hour gradient cool, as well as a stepwise cooling rate that kind of mimics PCR reactions, where we go from 95 degrees down to 60 degrees very quickly, and then slowly cool back down to 4 degrees. And the way that we test whether or not these different cooling rates are working is by running native page gels. So here's some of the results from those cooling experiments. On the left we have the 4-hour gradient cooling, and on the right the 2-hour gradient cooling. And each of these show 3 different bands. The top band being the fully formed monomer, the middle band being the partially formed monomer, in which 2 pieces of DNA came together but not a third, and the bottom band representing the unformed monomer, in which it just didn't work at all. And as you can see, just upon visual inspection, the right-hand gel, which is the quick 2-hour gradient cooling, actually worked a lot better because we have this much darker fully formed monomer band compared to the much lighter partially formed and unformed bands. And then we actually quantified those results using image analysis just to make sure, see just how much darker the fully formed band is compared to the partially formed and unformed bands. So after that, and being able to actually make the monomers, the next step was to make the hydrogel itself. So in order to visualize the gel, what we did was we formed it by placing the monomers and the aptamer strands together in solution with these little fluorescent beads so that they would incorporate into the gel. And then, this is just the stereo microscope image of the gel, and it kind of looks like a gel, but you can't really be sure from the picture. So what we did is we took the hydrogel with the fluorescent beads in it and put it into these special little microscope slides we made by cutting wells inside silicon gel. And so this is actually just a layer of silicon. It's got these little wells that we cut into it, and this gel actually goes inside these little wells. And then we place it under a fluorescence microscope and image the gel. We get pictures like this, and you can see the little beads present in the gel, and we use an image tracking program to monitor their progress through the gel. And so from these results, we got graphs like this, which show our control liquid, where the bead pretty much moves in a kind of random Brownian motion throughout the gel. And you can see here's the origin where all the beads start, and they just kind of go out in whatever direction they feel like. And so when we actually bring in the aptamer strands to make the gel, we get this, which is a very good result, showing that the beads move in a much more linear direction and a lot less randomly, showing that they're being trapped by the gel and the gel's actually being formed. So this result kind of gave us another proof of concept that the hydrogel is actually forming as we hoped it would. And these results, coupled with the super sandwich results, kind of serve as an overall proof of concept for both systems, allowing us to move to the second phase of experiments, where we begin to optimize the system by altering the material structure, with the hopes that one day it will be able to be used as a viable drug delivery platform that's going to be much more sensitive to the various range of small molecules. At this point, I'd like to just make some acknowledgments. First to the Plaxco group, to my PI, Dr. Kevin Plaxco, my mentor, Anna Simon, and the lab alumni, Camille Lawrence, for all their help with this project, as well as from the RAC group, Adam Pollock and Clayton Woodcock, who helped with the gel formation, the native pellage gels, as well as the Parsons group and the Materials Research Lab for the use of their equipment, Arika, Maria, Kevin, and Dr. Galen Stuckey for being the program coordinators, and finally to the Beckman Foundation and to the NIH for funding opportunities for this project.